All right, you guys, here we go. Another video here with on genetic disorders. Okay, and so this one, I hope you guys have fun with this. I had another act two that we would do normally in the classroom. However, since we're not in the classroom, I've brought it to you. Just kidding. Anyways, no. I've got a little alternative that we're going to do instead. Okay. And so let me pull this up really quickly. And then I've got a couple, I got a quick lecture I want to go through just to get you guys started to think about this, of what you could do here. So you guys, when we're looking at this, we're on this topic of meiosis. Sometimes errors do occur in meiosis. And when those errors occur, sometimes they can cause some different genetic disorders. And here are a list of some genetic disorders that can occur due to errors in meiosis. And I want you guys to explore these a little bit. You'll actually just pick one of these and create an infographic for it, giving me some key basic information about it, such as what is the disorder, how is it caused in meiosis, what's happening in meiosis doing that, and then how frequent is it that it occurs. And so there's a few like Pateau syndrome, Edwards syndromes, Down syndrome, you know that one, Klinefelter's, Jacobs, Triple X, Turner syndrome, and Creutzfeldt syndrome are just a list of a few that occur there, okay? So to get started, what I'm going to do is just quickly go over this lecture here. Let me get everything going where I need it. Can work? Come on. You can do it. It always does that. I don't know why. Okay, here we go. All right, so you guys, when we're looking at meiosis, we know, as you've done the lab already, that meiosis causes variation in the chromosomes number and structure, and it creates variation in the cells, and then sometimes we can get errors that would cause variation in the chromosome number, maybe. Someday I'll get used to this. All right, here we go. So close look, you guys, I want to do a little close look at the chromosome itself. So remember, we have 23 pairs of chromosomes in each one of our cells. And if we look at our chromosomes, they're actually broken up by genes. And so each section of a chromosome will code for a specific gene that will then code for a specific, it's a specific gene that codes for a specific protein that would then be expressed in our bodies, right? And so you can kind of look at this, all the different genes that are found on just this one chromosome in this one section, etc., that can be expressed. Now, again, remember, we have 23 pairs of these, so we have a lot of genes throughout that, and we actually have the whole genome mapped out, which is amazing. When we're looking at a chromosome is just tightly condensed DNA. So here's your DNA at the very basic level of just a double helical structure. And if you look at that closely, you can, if we looked, we could even see the base pairing, and we'll get into that a little later. But it's how it's read is what determines the gene, and then that gene would be then coded to make a specific protein. The other thing that you'll notice is that this DNA is tightly compacted around proteins called histones. Now, these histone proteins will then create supercoiling, is what it's called, tightly compact coiling of this to make your chromosome. And it's because it's so tightly compacted that we can actually see it with a microscope. And so if you were to try to see this right here in a microscope, good luck, right? It's just way too small. And we have so much DNA in our cells, it's just amazing. So when we're looking at karyotypes, you guys, I talked about this in a previous lecture for you guys, but you can see that each chromosome, that each pair of chromosomes, these are called homologous chromosomes. We can see that one came from the mother and the other one came from the father. And so it's a combination of both that cause this to occur. And so we get one half from mom, one half from dad. They form together to make that one organism, that offspring. If you notice, there's always just two of each type. You guys see that? There's never more than that. Well, there is, right? And so if we look at this, there's some things that can occur. One thing that can occur with chromosomes is we can have some mutations. And some mutations that can occur are such as deletions, maybe during meiosis, during that replication phase, or during crossing over. Maybe it loses a section of DNA, a whole section of the chromosome, and so several genes are then lost in that chromosome. Sometimes we get duplication where maybe it doubles that and we get duplicates of that. Maybe we get inversion where it kind of flips it. Sometimes we can get reciprocal translocation where we get this, and this is something that happens quite regularly, or it doesn't happen quite regularly. Sorry, don't get this mixed up with crossing over, because crossing over would be crossing between homologous pairs. This is not a homologous pair with this. Okay, and so sometimes that does occur though, where they match up during prophase 1, during crossing over, and maybe the two sets of chromosomes were not supposed to mix up and mix DNA. Okay, but sometimes that happens. It's called reciprocal translocation. Sometimes we get non-reciprocal translocation as well. Okay, and so here are a few different types of mutations that can cause genetic variation in the offspring. One that I really want to spend some time on is this right here, and we call it aneuploidy. And so what it does is it causes an imbalance in gene expression that is often detrimental to the phenotype of the individual. Usually it will cause a miscarriage of that child. Okay. There are two commonly types of aneuploidy. We can get monosomy or trisomy. Monosomy would mean that you're missing a whole chromosome. Trisomy would mean that you have an extra of that chromosome. And so when we look at this monosomy, you can see it's missing this right there, while trisomy, it's got an extra one. Okay. And so we're going to look at how this happens a little bit here, and you guys will be able to explore it a lot more in this activity. So what causes aneuploidy? Well, mostly it's non-disjunction during meiosis 1. Okay. And it actually can happen during meiosis 2 as well. Let's put that up there. Okay, so here's an example of this, of non-disjunction that's happening. This is showing it in mitosis, but the same theory applies in meiosis as well. So you can see that during metaphase, they should be lining up, and then during anaphase, these should separate out. But sometimes maybe this spindle fiber right here did not make it to the kinetochore of the chromosome to pull apart. And so now you can see that this cell now has an extra chromosome. It did not split. And so then we see this cell right here has missing DNA, while this one has an extra chunk of DNA that it does not need. And so when we're looking at this, here's meiosis right here. So this is showing it in meiosis, non-disjunction that can occur in meiosis. And so in meiosis 1, we can see that non-disjunction could occur. In meiosis 2, it can happen as well, the same process. Again, leaving cells that are normal. You got one that's got an extra one, and then you got this one that's missing one. Okay? Non-disjunction. Gosh dang, that stupid thing. So here are some examples of aneuploidy, and you guys will be able to research some of these. So I had you guys, one of the options was Down syndrome. So these are some different types. So you have trisomy 21. This is actually Down syndrome right here. You have Edwards syndrome, which is trisomy 18, and then you have Pateau syndrome, which is trisomy 13. And what it ultimately is for these three examples, they have an extra 13 chromosome, an extra 8 or an 18 chromosome, or an extra 21 chromosome. And you can see which one is much more prevalent, and it's trisomy 21. Okay, and it's Down syndrome for that one. What they've actually found is that the age of the mother it directly correlates to the prevalence of Down syndrome. So as the mother gets older through age, there's a higher chance in all ethnicities, not just Mexican or African American or non-Hispanics, and even so in all ethnicities we see this similar kind of trend here, that as you get older, the mother gets older, the likelihood of having a child with Down syndrome drastically increases. So anyways, just an interesting tidbit there when it comes to Down syndrome. Here's some other ones right here. Let me move that again. This thing's always in the way. Sometimes, you guys, we can get aneuploidy that's occurring in with the sex chromosome. So at that 23rd pair, we have an XX or an XY. Sometimes, excuse me, non-disjunction can occur with those. So for example, some males can actually have an extra X chromosome there, and we call this Klinefelters. Okay. Sometimes maybe they got an extra Y. That's Jacob syndrome. Females can actually get an extra X chromosome. It's called triple X. This isn't hugely detrimental to that female, because females actually already, since they have two of them, they actually shut down one already, and so they end up just kind of shutting that one down too. However, there is a little bit of a estrogen and other hormonal increase and decrease and whatever imbalance, and so it does cause irregularities in the menstrual cycle and other things like that. Sometimes the female might not get an extra, it only gets one X chromosome, maybe, so this would be monosomy, so it lost that extra X. It was fertilized with with an egg or sperm that did not receive an X or Y. Okay, and if that's the case, these are considered female. It's called Turner syndrome, and these are some characteristics that they notice there. Okay, all right, so I'm going to end right there, and so then go back to this right here. So what you guys again will do before I end up is I want you guys to go through, pick one of these that you're interested in researching. Look and see, how does this occur? How did errors in meiosis cause this genetic variation? All right. And then when you're done, you will submit your infographic to me on this assignment. All right. I'll talk to you guys later.